In this module, you will learn about the reproductive parts of the flower. What are the reproductive parts of a flower? In angiosperms, the reproductive parts are located in the flower. A flower comprises of sepals, petals, stamens, and carpels. Stamens and carpels are the reproductive parts of a flower which contain germ cells. Germ cells help in sexual reproduction. Do all flowers have stamens and carpels? Flowers can be bisexual as well as unisexual. A unisexual flower contains either stamens or carpels. For example, papaya and watermelon are unisexual flowers. A bisexual flower contains both stamens and carpels. For example, hibiscus and mustard flowers are bisexual. What is the main reproductive part of a flower? Stamen is the main reproductive part of a flower. It consists of the filament and anther. The filament is the stalk of the anther. The anther produces yellowish pollen grains from the pollen sacs inside it. These pollen grains contain the main reproductive cell or the germ cell. This germ cell involves in fertilization. What is the female part of a flower? Carpel or pistil is the female part of a flower. It is located in the center of a flower and comprises of three parts: the ovary, the style, and the stigma. The terminal part of the carpel is the stigma. It is sticky in nature. The stigma traps these pollen grains in its sticky surface. Then they can only travel down to the ovary for fertilization through pollen tube. What is style? The middle elongated part of the carpel is the style, and this swollen part at the bottom is the ovary. The ovary contains ovules, and each ovule has an egg cell. The pollen should fuse with the egg cell to produce the offspring. In this module, you learned that the reproductive parts of angiosperms, like flowering plants, are located in the flower. The stamen is the main reproductive part, and it is made up of two parts: filament and anther. Anther produces pollen grains. Carpel is the female reproductive part, and it is made of three parts: the ovary, the style, and the stigma.
And post-fertilization events. In flowering plants, pollination is an important mechanism that aids in transferring the pollen released from the anther to the stigma of the flower. If the pollen is of the right type, the stigma induces it to germinate and produce a pollen tube. The pollen tube, containing the two male gametes, then secretes hydrolytic enzymes which help the pollen tube to digest the tissues of the style as it travels down to enter the ovule through the micropipe, a minute opening in the ovule. Inside the ovule, the pollen tube encounters the embryo sac, the female gametophyte containing 3 antibiotics, 2 synogyns, an egg cell, and a central cell with 2 polar nuclei. The pollen tube now enters one of the synogyns through the filiform apparatus, during which it ruptures and releases the male gametes into the synogyn's cytoplasm. Of the 2 male gametes, one enters the egg cell and fuses with its nucleus. This fusion, known as syngamy or first fertilization, results in the formation of a diploid zygote. Meanwhile, the other male gamete moves towards the 2 polar nuclei and fertilizes them to produce a triploid primary endosperm nucleus or PEN. Incidentally, this fusion is known as triple fusion, as it involves the fusion of 3 haploid nuclei. After the triple fusion, the central cell is called the primary endosperm cell. Did you know that syngamy and triple fusion together represent double fertilization? A phenomenon unique to flowering plants. Double fertilization is followed by several post-fertilization events, which eventually transform the ovule into a seed and the ovary into a fruit. These events include degeneration of the antibiotics, synogyns, and the development of the zygote into an embryo and the primary endosperm cell into the endosperm by repeated mitotic divisions. Endospermic development is of 3 types: nuclear endospermic development, cellular endospermic development, and halobial endospermic development. In nuclear endospermic development, which is the most common type of endosperm development, the primary endosperm nucleus divides mitotically and forms 3 nuclei. This division is sometimes followed by cytoplasmic divisions from the periphery. In the case of cellular endospermic development, which is seen in plants such as Utricularia, the division of the primary endosperm nucleus is immediately followed by cytoplasmic divisions. Halobial endospermic development, on the other hand, is an intermediate type of development where half the cells develop in cellular mode and the other half undergo 3 nuclear divisions. Interestingly, the number of free nuclei formed prior and post-cellularization differs greatly. Take the case of a tender coconut fruit, where the coconut water that you see is actually thousands of free nuclei. As the coconut matures, cellularization leads to a decrease in the quantity of water and the formation of the white kernel, the cellular endosperm. Therefore, in the coconut, we find both free nuclear endosperm made up of thousands of nuclei and cellular endosperm. Histological studies have shown that endosperm contains stored food in the form of starch or proteins, as in cereals or oil, as in castor or coconut. This food provides nourishment to the zygote while it is developing into an embryo. It is interesting to note that the endosperm also forms a major source of nutrition for human beings. Incidentally, the development of the embryo, also known as embryogeny, occurs only after a certain amount of the endosperm has developed. In fact, in some plants, such as groundnut that belongs to the bean family, the mature seeds lack endosperm as it gets completely absorbed by the developing embryo. When these seeds germinate, the cotyledons or seed leaves become the source of nutrition for the embryo. However, in mature seeds of castor, we see that the endosperm is retained as a storage tissue. In the case of cereals, the annulum layer, which forms a part of the endosperm, stores food as well as secretes amylase enzyme for the digestion of stored starch during germination of the seed. Studies by botanists have revealed that during early embryogeny, the zygote divides into two cells. The basal suspensor cell towards the micropylar end and the terminal embryo cell towards the chalazo end. The basal suspensor cell further undergoes mitotic divisions and forms 4 to 8 cells which push the terminal embryo cell into the tissue of the endosperm to allow it to derive nourishment. Moreover, the terminal embryo cell also undergoes divisions to form a group of cells known as proembryo, which subsequently develops into a globular, heart-shaped and torpedo-shaped mature embryo. The structure of a mature dicotyledonous embryo is nonetheless different from that of a monocotyledonous embryo. A dicotyledonous embryo, for instance, consists of an embryonal axis and 2 cotyledons. The epicotyle, the portion of the embryonal axis above the level of the cotyledons, terminates with a plumule or stem tip. Below the level of the cotyledons is a cylindrical portion called the hypocotyle that terminates at the radical or root tip, which is covered with the root cap. On the other hand, a monocotyledonous embryo, commonly found in the grass family, has only one cotyledon called a scutellum, situated towards the lateral side of the embryonal axis. The upper portion of this axis, known as the epicotyle, lies above the attachment of the scutellum. The epicotyle has a shoot apex and a few leaf primordia enclosed in the coleoptile, a hollow foliar structure. The lower end of the embryonal axis has the radical and root cap covered in an undifferentiated sheath called the coleorhiza. Interestingly, the various parts of the embryo develop differently as the seed germinates. In this manner, the embryo, as well as endosperm, develops from the zygote and primary endosperm cell after fertilization has taken place in flowering plants. You have now reached the end of this module. In this module, you learnt that singly or first fertilization involves the fusion of one male gamete with the egg cell present in the egg apparatus that results in the formation of a diploid zygote. Degeneration of the actividals and synergids, development of the zygote into an embryo, development of the central cell into primary endosperm cell. The primary endosperm nucleus undergoes 3 nuclear divisions to form 3 nuclei, which develop into endosperm following the formation of cell walls. The main function of endosperm is to provide nourishment to the growing embryo. The development of the embryo is called embryology.
In this module, you will learn about the history of cell division. Our Earth is a beautiful place in which various types of organisms coexist. From simplest life forms like microscopic bacteria to complex organisms like huge blue whales, all have a basic structural and functional unit called a cell. Did you ever wonder how the cells evolved into an organism? The investigations made so far make it clear that all living beings are made of cells and that each cell has a prominent nucleus. Matthias Jacobs Claden, a botanist, investigated about plant cells, and Theodor Schwann, a zoologist, investigated about animal cells. Around 1838-39, Claden and Schwann together formulated the cell theory, which is applicable to all living organisms. However, this theory failed to explain how new cells originated in living organisms. In 1855, Rudolf Virchow modified the cell theory formulated by Claden and Schwann. He proposed a phrase, omnis cellula decellula. Somes carry inheritable or genetic elements. During mitosis, each somatic parent cell divided into 2 daughter cells having the same number of chromosomes as that of the parent cell and maintained their constant number. At that time, biologists wondered how the progeny two maintained their constant chromosomal number. Further investigations of cell division put forward the concept of meiosis, which was discovered and described by German biologist Oskar Hertwig in 1876. During meiosis, the number of chromosomes in the parent reproductive cell is reduced to half the number in male and female gametes. When these haploid gametes fused during fertilization, the progeny maintained the diploid chromosomal number, similar to the parent. Let us see what happens if all reproductive cells are not undergoing meiosis, but only mitosis. In that scenario, parent reproductive cells having 46 chromosomes will share the same number of chromosomes to the gametes, thereby leading to 46 chromosomes in each. During fertilization, when these gametes fuse to form a zygote, their chromosomal number will be doubled to 92. If mitosis continues further in first and second progeny and so on, the number of chromosomes will be doubled to 184 368, and so on. But this is not happening in successive generations. In 1890, August Weismann, a biologist, hypothesized that in successive generations, all the individuals of the same species maintain same number of chromosomes at the end of each cell division. After a series of experiments over the next 50 years, the chemical nature of hereditary material in chromosomes was finally determined to be deoxyribonucleic acid, that is DNA, by Hershey and Chase. In 1953, the double helical structure of DNA was discovered by James Watson and Francis Crick. These discoveries on cell division made the scientists explore cell biology further, which led to a revolutionization of medicine and agriculture. You have now reached the end of this module. In this module, you learn that all organisms have a basic structural and functional unit called cell division. Walter Fleming named the process mitosis, and later the complete process of mitotic division was illustrated by Theodore Boveri. Waldeyer named the thread-like structures chromosomes, which means colored bodies. Wilhelm Roux proposed that chromosomes contain hereditary material. Hugo de Vries, Carl Eric Correns, Erich von Schermach researched independently and rediscovered Mendel's laws and concluded that chromosomes carries inheritable or genetic elements. During mitosis, each somatic parent cell divided into productive cell is reduced to half the number in male and female gametes. The chemical nature of genetic material was finally determined to be deoxyribonucleic acid, that is DNA, by Hershey and Chase. The double helical structure of DNA was discovered by James Watson and Francis Crick.